how thrilled we all are this day is actually happening. We'd planned to have the launch several times, but because of the virus, we had to postpone. So we've gone the technical route by doing this online, and I think it's quite an exciting development. There's so many people we have to be grateful to for all the help they've given us. And in his absence, my namesake Colin Williams and his wife Sue have been a mainstay of the development of the group, providing fantastic admin support and keeping us in check. Eric is, from a standing start, cultivated the website, and so many people have commented on how effective and impressive that is. And we hope it'll be a tool for you to use to inform us about what you think should be happening, but also for us to communicate back to you developments in the field and to broaden the involvement of people in research and to make people aware of what's going on. I've said this a number of times over the last year. We're so lucky to have Liz and Chloe from Parkinson's as our staff support team. They're incredibly committed, so helpful, and we bonded as a team. It's a lovely partnership between Parkinson's and the group. I'm delighted that David, who has only recently joined the group, has agreed to take over the administrative function, which is an enormous help. And also he's been involved in the website development. So there's a lovely wee team developing of enthusiastic people who are committed to what we're about. And it's exciting development today to have this launch. We've been helped by so many people external to Parkinson's at Glasgow Cali and Andy over in Strathclyde. And we felt so welcome by the members when we joined them at meetings and been at conferences and so on. And Annie McLeod, who will be speaking later, has provided a lovely welcome as the director for Scotland. I think for now that's probably enough of introductions, other than say we are absolutely thrilled that Dr. Donald Grosset, who has invited us to call him Donald and keep this all informal, has agreed to be our first speaker this afternoon. His reputation is wide and quite so impressive that we're really proud to have him as our main support today. Donald, I want to say a little about his work, but he's consultant neurologist at the Institute of Neurological Sciences, honorary professor, Institute of Neuroscience and Psychology in Glasgow. I'm going to be speaking about the Tracking Parkinson's Research Study, which is the world's largest long-term study in Parkinson's, and we'll take questions and answers about that later. So I think for now, Liz, is to say welcome to everyone. We look forward to your questions. I hope they're fairly robust. I hope some are fun. And to launch the start of the Greater Glasgow Research Interest Group, can you please welcome with me Dr. Donald Grosset as our keynote speaker. Donald, over to you.
Thanks very much, Colin. I'm delighted to speak to the group. This is I've done a lot of Zoom and other platforms in the past 6 months, and this is the biggest Zoom meeting I've done. So I'm very impressed by the number of people who are signed in today for this launch meeting. And I had a quick look as people were joining, and I see some names I know. So hello to those I know. Hello to everyone. So as Colin said, I'm going to speak about the Tracking Parkinson's study. And I'm going to link that to our current thinking about how this kind of work will influence treatments for Parkinson's in the future, will help to bring new treatments to Parkinson's. So what I'm going to do is share my screen. So the Tracking Parkinson's study is a UK-wide study, and in 2010 we got started with this project. So it's 10 years old, and It brought together a number of universities with various disciplines, epidemiology and neurology and genetics. And it's very much a British project. We're very proud of the fact that it's a Scottish project and run out of Glasgow University. But it wouldn't have been possible without all of the people listed here. And our laboratory work is done in Wales, and a lot of the analysis work has been done down in different parts of London. So a big British effort here. As Colin said, it is the largest study of Parkinson's ever conducted. And it's now getting to be one of the longest as well. So what this project did, and it involved in excess of 50 centres as shown on the map here, and was heavily supported by the UK Clinical Research Network, CRN for short, which is a less well-known branch of the NHS, but a very important one. So alongside the delivery of care, there's a network of nurses and scientists who are part of the NHS Clinical Research Network. That is part of the devolved arrangements, as healthcare is. So there are 4 different names there for the different devolved nations. But that is a backbone of, which supported our study from the outset and has been very important. And what we set out to do was to ask people with Parkinson's and their brothers and sisters to enroll in an observational study where we would record how Parkinson's was affecting those with Parkinson's and take blood samples looking at DNA from all participants and do long-term follow-up to understand the evolution of Parkinson's and how that is linked into genetic and some other elements. So what that meant was initially in the first 3 years of this project that we saw people every 6 months, and then we've extended that to every 18 months. And we recruited almost 2,000 people with Parkinson's. That's recent onset Parkinson's. We had a young onset Parkinson group as well, which was slightly smaller at about 300 people, and the brothers and sisters was around 450. Now, I sometimes get asked, why did we recruit brothers and sisters? Because we had an interest in genetics, as I will go on to explain, and because our brothers and sisters are the most genetically similar to us. So parents obviously have genetic similarity to us, but our brothers and sisters are the most genetically similar. And they are generally of a somewhat similar age as well. So they made sense as a comparator group to understand genetics and genetic variation, and some people with similar genetics not getting Parkinson's while others do. So the siblings, the brothers and sisters, were not to have Parkinson's at entry to the project as a kind of control group. And a number of observations were made, and many observations were repeated, looking at changes over time, and blood specimens were taken repeatedly every 18 months. Only once for DNA at the beginning, because the DNA doesn't change, but taking a blood sample and storing it in the freezer for additional studies, which I'll make brief mention of as well. So we invited people to join the study, and we're delighted to recruit in quite a short time for such a huge project, a little over 2 years. And you'll see the average age at entry there, and the usual split of male to female, 2 to 1 male to female, which is the usual. And this recent onset group had to be diagnosed within the preceding 3 years the earlier the better for entering the project, and the average was 1.3 years after diagnosis that they entered the project. So by between August 19 and around about November 2021, people are at 7.5 years duration in the project. The project currently is scheduled to run through to April 2022, and it's not yet decided what to do at that time, whether to continue on or not at that time. Now, the concept here was to collect a lot of information about treatment for Parkinson's and other things, about any other health problems, all about the symptoms and the signs that we pick up on examination. So looking at the classic Parkinson features, the tremor and the stiffness and the slowness, and scoring the severity of that. Examining sense of smell, since that is affected in most people with Parkinson's. Doing these serial blood samples, as I've mentioned, and asking participants to fill out questionnaires which address different components of how Parkinson's might affect them, such as the tremor or their sleep or anxiety, a whole range of features that we wanted to understand in considerable detail. So this was quite a commitment by the participants to do this and to be willing to keep repeating these questionnaires, which were generally every 18 months, and people have been outstanding in their contribution to this, and giving the blood samples repeatedly as well. So we have a vast array of information. Now, like any study, it has to have aims, and what we had observed in routine clinical practice was that there was a variation in Parkinson's. Everybody knows this, and everyone with Parkinson's knows this, that one person's not the same as the next. And yet there's relatively little work being done to look at why there is this variation and how it comes about. And I've highlighted 4 key features here that we wanted to look at. One was how treatment responsive people are. The next was the progression rate. Some people, fortunately, Parkinson's really never progresses rapidly, but some people are very slow and some are more fast, and why the variation. The non-motor features, so some of these other elements that I've talked about, sometimes the bowels get a bit sluggish or the waterworks are problematic. I've mentioned sleep, anxiety, depression, whole host of non-motor features that vary, and some people will get that as a predominant set of problems, while others might have a predominant motor problem with tremor, for example. And then cognition, we do know that cognition, memory, and other thinking processes can decline with Parkinson's, but not in everyone. And usually after many, many years of Parkinson's, but again, there's variation in that. So we wanted to use all of this information to, first of all, define that variation, and secondly, explain it. And to link that, and I've mentioned that we took blood samples for DNA, to link that to genetics. And then we would, as well as doing this project and banking all this information and analyzing it ourselves, we would also make that information and the blood samples available to other researchers worldwide if they had a clinical project that would be benefited by accessing this big data set. And the idea here is to say that we want to improve treatments for Parkinson's, but no longer consider Parkinson's as one entity where treatments will all work for everyone. There may be treatments that will work for one subtype of Parkinson's and not another. And if we understand that subgrouping, we will be more successful in targeting the right treatment for the right subtype of Parkinson's. And this is very important in designing and interpreting drug treatment studies, clinical trials, Because if we have a treatment that might help a quarter of people with Parkinson's, but we give it to everyone, we might not see the benefit because we've only got a quarter of the people in the sample who would benefit. So these are the thinking processes behind this study. I'm going to talk a little bit more about treatment responsiveness. The classic definition of Parkinson's says that there is an excellent response to medication. It's L-DOPA based, and many of you will know that as Zinimet, Madopar, cocarodopa, cobeneldopa. The L-DOPA is the active ingredient in all of these, and almost everyone with Parkinson's will go on an L-DOPA based preparation at some point in their time with Parkinson's. A looser term than Parkinson's disease is Parkinsonism. And this means that there's a kind of appearance that mimics Parkinson's disease, but it isn't the true condition. And these other conditions can have generally a more severe effect on people. They're more severe and more rapidly progressive. And part of that is because they have a poor treatment response to the L-DOPA. So that's the classic distinction. And yet, but before I go to the and yet, and the criteria for Parkinson's, which are the classic UK brain bank criteria that came from Queen Square, again from a national project funded way back by Parkinson's UK when it was called the Parkinson's Disease Society. Here are these classic definitions: an excellent response to levodopa on the supportive criteria, and an exclusion criteria, a negative response to large doses of levodopa. So what we wanted to do was, in a way, to challenge this and say, does everybody with Parkinson's have an excellent response? Or putting it another way, is there a middle ground between excellent and negative? How common is the response less than this excellent response, which is in the criteria for Parkinson's, but positive? You know, it's not a negative response to large doses. It's a middle of the road response. And what effect does that have? If there is that response variation, what effect does that have on Parkinson's, how it progresses through time? And this is what we did. We asked people to take their usual morning dose of L-DOPA-based medication, and we measured what they were like before they were taking that dose to after, and they ought to improve. And we can measure that using the scale, which is on the y-axis, the UPDRS scale, which measures, it's a summation of the motor features, the tremor, the stiffness, and the slowness, and looks at different body parts and looks at walking and voice and all the kind of classic features of Parkinson's, and says before treatment, actually having missed out treatment from the evening before, what was the person like and what percentage improvement did they have after they took their usual morning dose? And this, in the bottom left, you'll see this is 1,007 cases out of our nearly 2,000 cases. At the point of doing this response test to L-DOPA, the patient had to be on L-DOPA treatment, which not everyone is. So it was 1,007 cases, or just about half of the people participating in the project. And this actually is bigger. This single observation in these 1,000 people is bigger than the whole of the rest of the world literature put together that came before it. So this is massive in scale compared to anything that had been done before. Now, what it did was it split people into those with a limited response and a definite response. And these people all fulfill these criteria for having Parkinson's disease that I've mentioned. And yet there are some with a more limited response and some with a much more definite response. And The proportion of cases is shown there. It's 39% have a more limited response and 61% have a more definite response. So what were the differences? Well, those with a definite response were a little bit younger. They had milder motor features. And on that scale, the UPDRS scale, it's 3.7 points, which is that's a relevant number. That means that there's a bit more of tremor, stiffness, slowness, or any mix of the three. And even more importantly, they didn't progress so quickly. So looking at an 18-month period, there was again a slower rate of progression when there was a good treatment response, which sort of makes sense that if you're responding to treatment, your dose might go up over time, but if you're responding to treatment, then it will keep the symptoms under better control. But no one had ever really analyzed it in this way before, and certainly not at this scale at all. So I mentioned 18 months, and now on the x-axis, we've got it in years. So the 0 to 18 months is 0 to 1.5 years in the study, and we've gone on and looked at progression over time out to 4.5 years so far. And what we've got is the 1,007 cases split again into limited and definite as regards their response. And we can see that those with a definite response have a lower score, so that's less severe motor features. They've got a lower score at baseline than those with a limited response. And they also have a flatter curve over time, right out to the 4.5 years. So just annotating that, it kind of shows the difference, that there's a little bit of a difference at baseline and even more of a difference at the 4.5 years of follow-up. And in fact, those with the definite response are only marginally worse than those, are only marginally worse at 4.5 years than those with a limited response are at baseline. So this makes a huge difference to how their Parkinson's behaves over time. So the other defining feature about Parkinson's that so many of you will be more than aware of is that it's not a static condition, it fluctuates. And it's almost a defining feature. It might not be so evident in the first few years, but over time, this fluctuation from being good when you're good and bad when you're not so good, when you're wearing off, is a key feature of Parkinson's. And we can analyze this. And there was a questionnaire developed for this called the wearing off questionnaire. There are different versions of this, but this is the one that most people use. So it's 9 items, and you will see that it covers both the motor symptoms that I've been referring to and some of these other non-motor symptoms, some of which I've mentioned, like anxiety and mood, but also pain and slowness of thinking, so a kind of cognitive slowness. So when treatment is working well, these symptoms, both motor and non-motor, should improve, and then there can be this wearing off, sometimes between doses, particularly as the years go by with the Parkinson's. So the first thing we do is ask people, do you get these symptoms on a typical day in the past month? And we've got our 5 motor symptoms on the left and our 4 non-motor symptoms on the right, and the proportion of people with this mix of motor and non-motor features. And we can see that motor features are more prevalent. The percentage of cases on average, mid-60s, and about 40% of people on average have a non-motor symptom, and a little bit of variation, the commonest in the non-motor being pain and aching. And this was in 1,500 cases recorded at 4 years of Parkinson's, 4 years after diagnosis. So then we can say, well, you've got those symptoms on a typical day in the past month. Do they improve when you take your dose of medication? And then we've split it into yes in green on the left and no in red on the right. And we can see just visually that the motor symptoms improve to a greater degree than the non-motor symptoms. There is improvement in some people for all of these different components. Motor generally more responsive than non-motor, but not an overwhelming yes for response, even to something like stiffness and slowness of movement, being a little over half, something around the 55% of people who say, well, I do have that symptom and it does improve with my next dose of treatment. So this is informative to our thinking, because I hear eminent people at times, eminent neurologists saying, well, we're very good at treating the motor symptoms of Parkinson's. We don't need to worry about that because we've got all of that sorted. This is telling us the real story, and that we do need to look at new treatments that address the deficiencies that we're highlighting here in the ability to improve symptoms with our treatment. So what we wanted to do in thinking of this subgrouping approach is to say, on the top right here, we're saying, look at all these different colors. There's variation in people with Parkinson's in all of these motor features, these non-motor features, the degree of motor responsiveness, whether there's any trouble with memory, and so on. So can we look at people at baseline with their Parkinson's and examine their motor features and their non-motor features and their treatment response? And then can we say, on the basis of what the person has at the initial diagnosis, can we predict the future? Can we say what's going to happen as regards progression? So we set out to do this with our epidemiology and statistics colleagues with all of this data. And we've come up with this subgrouping. And the colors here are traffic light colors. So we have got 4 groups. We've got, on the bottom quadrant there, we've got one group who are actually much better than average regarding their motor, non-motor features and their treatment response, but their progression is middling, reflected by the orange. And there are 3 other groups. So let's look at, that's the concept. Let's look at the actual groups and what clusters together. So this is, in statistical terms, referred to as a cluster analysis. And it takes an enormous amount of data and puts it into a computer analysis program, rather than us trying to sit and work out the connections between the different components. So what we have is the 4 clusters. They're roughly in quarters, not exactly, but cluster 1 has a faster motor progression and has some other features. Prominent hyposmia, that's loss of sense of smell. Hypotension, that's low blood pressure. More prominent cognitive impairment. Not such a good dopa response. And unfortunately, the fastest motor progression out of the group, out of the overall group. I've mentioned the better than average cases with this intermediate response. That's cluster 2, mild motor and non-motor. Cluster 3 is more severe on the motor side, and poor sleep and poor general sense of well-being. And you can see in the outer baseline motor that they end up needing more medication and also have more gait and posture problems. And then cluster 4 actually have a slow motor progression, but a poor treatment response, which sounds strange from what I've said earlier, but in fact, this is what we recognize in the clinic already, that some people are what we call tremor dominant. So their tremor is really bothersome, but they don't get very much problems in any other sphere. So it's tremor dominant, but missing some of the other problems. And tremor does not respond as well to levodopa, which explains that issue. And it tends to not progress so fast. So the tremor can persist or even sometimes burn out and improve, not due to treatment, just due to some kind of brain resetting. So we have 4 groups here that are significantly different one from another. And just putting some more data on that, cluster 1 then, the faster motor progression, they're the oldest of the 4 groups at 68 years, and cluster 2, mild motor and non-motor, are the youngest at 62 years. And the Doppler response, poorest in the top left cluster 4 at 29%, and best in the younger cluster 2 group at 36%. So these are substantial differences in responsiveness and in age. So that's the cluster analysis that we've done. And that is very informative as we think about people entering new treatment trials that are designed to try and slow progression. If we can predict what their progression should be, then we can much better get a handle on how well the new treatments are working. So I've mentioned genetics a few times, and I'm going to talk to you first of all about classic gene mutations. Now here I've taken our 2,000 or so people with Parkinson's of recent onset, and then our young onset Parkinson cases as well. So now we total 2,262 participants, and out of that only 29 or 1.4% had mutations in the genes. And there's details there of which those genes are. And other studies have shown this before. This is not a novel observation particularly, but we've defined it in a kind of large British group, which is important as we take forward treatment approaches targeting genetics in Parkinson's disease. When we look at people who have very young onset Parkinson's, which is fortunately rare, in fact, the mutation rate is considerably higher at 18%. So against this overall 1.4%, it's 18% at very young onset. And when we take people under 50, so youngish onset, then 3.3% have got these different mutations in these 2 genes called Parkin and PINK1. If there is a family history of Parkinson's and what we call large dominant families, so that means that Parkinson's is running in the family in a dominant fashion, which means usually that 1 in 2 people are affected from one generation to the next. The mutations there, as you would expect, because it's a genetic pattern, are running at 7.4%. So what this suggests is that the genetic component for Parkinson's is much more relevant to younger people, and much more relevant when there is a strongly positive family history. Now that's important to know in the British setting, because the LRRK2, which I've given some information about on the bottom left there, we had only 18 cases, under 1% in our study. But in fact, in Arab populations and Ashkenazi Jewish descent populations, it's much more common. It's 40%. So treatments targeting LRRK2 are not going to be so relevant in the British setting. They will be relevant to a small proportion of people, but internationally, it can have significance in some populations. And that's relevant because treatments are being developed to target LRRK2 and the problem caused by the mutation. The K in LRRK2 is kinase, and there are drugs called kinase inhibitors, which are actually used at the moment already to treat people with rare forms of cancer. And these are being developed to target LRRK2 problems in people with Parkinson's. So I've mentioned dominant family history. The opposite of dominant in genetic terms is recessive, and that usually means that the parents are unaffected but are both carriers, and the mutation is carried in from one parent on each side and joins together to form the problem. And in our young onset people, actually dominant and recessive mutations were equally common. We didn't see differences between people with LRRK2 and people who did not have LRRK2. We did see difference with these rarer Parkin and PINK1 mutations with more posture symptoms but less cognitive impairment. And we found that people with Parkin had a preserved sense of smell, which is would be unusual or much more unusual in people without such Parkin mutations. And we can project, because of the scale of our study, we can project that there are in fact 1,000 people in the UK with a genetic mutation as a cause of their Parkinson's. But you might correctly think that's not an awful lot, because didn't I hear that there's 140,000 people with Parkinson's in the UK? So 1,000 people out of 140, one in 140, that doesn't sound very many or not relevant to all that many people. Well, the thinking about genetics has gone beyond the genetic mutation to this other phenomenon, which I will now mention, which is genetic variation. And this variation is based around different patterns seen. It's not a full mutation, but it's a variation in a smaller segment of DNA than would cause a mutation. So let's talk about genetic variation and whether that's relevant to more people. And the main genetic, these are called genetic risk variants rather than mutations. And the main one that has evolved is this GBA. And I'm going to give you a little bit of a story here about this young boy called Brian Berman. And you see that the arrow points to the fact that his tummy is distended, and also, in fact, there's in the yellow arrow, there's a syringe going in there delivering a treatment to him. And he has this rare condition called Gaucher's disease. And it turns out that when a person gets two of the GBA variants, one inherited from each parent, they have Gaucher's, causing this swelling of the stomach. It's actually primarily the liver that swells. But the carrier of GBA, of a single abnormal GBA, in this case the father and mother, are at risk of developing Parkinson's. So on the picture here, who's at increased risk of Parkinson's? Well, the parents certainly, because they've got carrying this single variant of GBA, and possibly the two brothers, a 50% chance that they will carry that gene. They don't have the Gaucher's disease, but they might be a carrier of the gene, and they are at risk of developing Parkinson's. And there is a whole treatment program around underway in terms of targeting GBA in people with Parkinson's. You can see the statement there, GBA mutations are the most prevalent genetic risk factor. So these figures and facts are from Sanofi, who are one of the companies developing a treatment to target this. And they make this statement, overall GBA, they call it mutations, but it's really variation. Overall GBA variation has a prevalence of 5% in people with Parkinson's. So what we did in the Tracking Parkinson's study was set about to examine whether that 5% figure was true. I've already commented on the difference in different populations for the LRRK2, which is much rarer. What did we find with the Tracking Parkinson's study? Oh, incidentally, before I come to that, the little lad with the swollen stomach has become the chief executive officer of the National Gaucher Foundation in the US, and got pioneering treatment, which he was the first in the world to receive pioneering treatment, which was back in his childhood, and it's given him a normal life. And he's then developed this National Gaucher Foundation. So a big success story in targeting GBA in a different disease process, but raising promise of something similar being possible for people with Parkinson's and the GBA variation. So what we found was, and the big red arrow points to our figure, what we found was a 9% rate. So actually higher than quoted by Sanofi in the UK population, a total of 9%. And the reason that we've got this higher figure is that this gene is actually very difficult to sequence and requires an enormous amount of work to get the readout. There's been huge progress in analyzing DNA, and it's become very automated in large measure, but GBA doesn't lend itself to that, just the nature of the gene and how it sits. So our laboratory colleagues in Cardiff spent many, many months analysing and got a readout of 9%, and looked at all the different subtypes which are in the table on the left there, which I'll not go into in any more detail, other than to say there's the 9% that are definitely relevant, what's called pathogenic variants, and then we've got another 1.4% in there with the big long list, which is called rare variants of unknown significance. These are probably relevant as well, but no one's ever described them before. So it's unknown significance until some other groups identify these as significant in people with Parkinson's. So many of these will likely enter the list of definite relevant GBA variants. So what did we find? We found that people who have these GBA variants are younger. So the genetic element is coming in and causing younger onset by around 5 years, that they tended to have more trouble with their posture and balance. They tended to be more advanced in their motor score, that's the Hoehn-Yahr scaling that's used for that. But at entry to the study, they had no difference in their cognition. Now this is relevant because some smaller work done before has suggested that people with GBA are more likely to have memory trouble. We did not find that at study entry. We will examine whether that is true over time in due course. So this is very much an area of active research interest globally, with more than one drug company. The top left shows from one of the Fox Foundation websites that there's a drug treatment there being developed. And this is targeting the GBA, which is linked to this protein alpha-synuclein, which we think is very central to the problem with Parkinson's in 99% of people. So coming back to what we're doing in tracking Parkinson's, as well as looking at these genetic mutations and genetic variations, we're combining all of that with all the other observations to look at genetic factors related to progression. And this is enormously complicated computing, which is highly specialized and which I couldn't do. And I have two colleagues in London who have done this. And what they've done for this, you need a lot of data. We have a big study, but we need even more data. And what they have done to get this kind of complexity of analysis and make sure it's accurate is to also collaborate with Oxford Discovery, who have a project with about 1,200 people with Parkinson's, and PPMI, which is an American study with about 400 people with Parkinson's, and add all of that together and look at the genetics relating to progression. Now, what they've done is analyzed all of that against motor progression on the left and cognitive progression on the right. And they look at the chromosomes, which is along the bottom on the x-axis. And we've got the classic 22 chromosomes there, and whether there are genes that link to this progression. And so far, they have found one linkage, which is to a gene called ApoE, which is relevant to the development of cognitive impairment, and actually also features in Alzheimer's disease, in a subset of people with Alzheimer's disease. This study despite the vast numbers of cases, this study is actually not quite big enough to do this kind of analysis, which is called genome-wide association study, or GWAS. So our aim is to get even more studies combined to this, to do this very sophisticated whole genome analysis against the clinical parameters in Parkinson's as time goes by. Now, I've mentioned alpha-synuclein, and I'm going to talk to you about alpha-synuclein as a protein, and a protein that can exist in different states. And that's why I've put in the eggs here, because it's a good comparison. So a raw egg can be cooked to varying degrees and end up as a hard-boiled egg. And alpha-synuclein can circulate around in the blood and the brain as little globules, or it can combine into little clumps. And then it can abnormally now, clumping is fine because it can unclump the two green arrows. So from little globules to little clumps is fine, but then it can form into these linear fibrils, and then even more troublesome, can cause these kind of big conjoins of all the alpha-synuclein fibrils. And that's been regarded as irreversible up to now. So similar kind of concept to what happens with proteins in an egg. And these fibrils are damaging to the cells and are present in 99% of people with Parkinson's in the cells that are deteriorating with Parkinson's in the brain. Now, the analysis of this, you wouldn't really think it was possible to uncook an egg, and yet this has been done recently, a process which is one would think of doubtful relevance to anything, has been invented, in fact, from Australia to unboil an egg. And this has applications in industry and other such, not unboiling eggs, but disconnecting proteins and reversing certain processes is an important, actually very valuable device called this vortex fluidic device. So in a similar vein, we have treatments now saying, let's target the clumpy protein. In Parkinson's, the protein is alpha-synuclein. In Alzheimer's, it's tau. Can we target these proteins that are clumping together and unboil them or disconnect them? And there are other conditions where alpha-synuclein is relevant, as listed on the left, some of the dementias, and this other Parkinson disorder called multiple system atrophy. And tau is relevant, as I've said, in Alzheimer's, but also another condition called progressive supranuclear palsy. And we have drug companies developing treatments targeting these. So there's an overlap here because in early Parkinson's, in fact, somebody can appear to have early Parkinson's and actually have one of the other conditions, the PSP in short, progressive supranuclear palsy. And then the cognitive impairment in Parkinson's, some cases will have Parkinson's and Alzheimer's. So there could be dual treatment used in the future for people who've got dual problems with each of these proteins. So I mentioned this approach of combining even more data than the 3 studies, and this comes from an analysis of 17 data sets, including our own. This now then amounts to 37,000 cases. And there's a comparator group here of 1.4 million controls, so our numbers have just gone hugely up. And I've been talking about 3 of the genes that are shown in the red arrows here, the GBA, relevant to that Gaucher disease, and the young lad who went on to set the foundation up. SNCA, which is the alpha-synuclein I've been talking about, LARC2, the one relevant more to Arab populations and Ashkenazi Jewish populations. And you can see that, in fact, there are 90 independent genetic signals now tied into Parkinson's. And that these explain something approaching a third of the risk of developing Parkinson's. Now, there's an important point here that there's, first of all, there's the risk of developing Parkinson's with a certain genetic pattern. And then there's the influence that the genetic pattern has on how the Parkinson's plays out over time. And these two are not necessarily the same thing. It's not necessarily the same to say you have LRRK2, therefore you have an increased risk of Parkinson's and you will progress more rapidly. It could be a completely different gene that's relevant to rapid progression. So our study with this 10-year duration is important to understanding the relationship with all these complex genes and progression over time. So I'm going to mention, finish with a kind of human element here, and it's relevant to the international scene as regards Parkinson's, and it ties into our own work. So on the top left is a picture of Eugenia Brin, who is the mother of Sergey Brin, who's pictured in the middle. Sergey Brin, co-founder of Google. And Eugenia has Parkinson's, and she's LRRK2 positive. And Sergey Brin is not got Parkinson's, but might be at risk of developing it. And because he is multi-millionaire, I think he must be a multi-billionaire by now, he has put multiple millions of dollars into a big program of research, which is unparalleled in ever. The degree of investment that has come from the Google millions or billions. And one project, and of course he's a data person, he knows how to handle data. So one project is to go to the next level in terms of combining all the data from these different studies. And we're in the process of setting up how we will contribute our data to this international collaborative project to take this all to the next stage. And in parallel with this, the drug companies who are interested in developing new treatments are aware of all of this and connected into us and know what they can tap into and how this will help them test their new treatments. And in fact, the Eugenia Brin's particular LRRK2 mutation is known about in detail in the spirit of openness that I suppose is the case in the US in terms of data sharing. So she has a particular mutation. And this other person with a young onset Parkinson's, we don't know her status, but she probably has a genetic type of Parkinson's. And this is Charlotte Johnson, who's Boris Johnson's mother. And she drew this picture at the age of 36, 4 years before she developed Parkinson's. And one can guess from this that she was beginning to have early features. And she's showing some hand that looks a bit menacing, dragging her along or telling her to come along. And she's showing how her hand is not working right and is got tremor and is being tied down. So she was diagnosed young with Parkinson's. And more recently, at least according to Tatler magazine, which is where this information came from, she had deep brain stimulation for her Parkinson's at the age of 75. So she's had a long lifetime with Parkinson's, and the recent brain surgery has actually helped enormously 35 years into having Parkinson's. These two cases, or three really, because there's Eugenia and then Sergey and then Charlotte, these are all probably all genetic types of Parkinson's. And they illustrate that we need to be thinking, which gene is relevant? Which pathway is this gene linked to? Is this gene linked to developing Parkinson's? Yes, in the cases I've shown. Is it linked to the time course of Parkinson's, how the features progress over time, or not? So this is about finding the right treatment for the right person, finding the target, but using science to find that target and then treating it. So I would like to thank Parkinson's UK for funding this project. They have been a fantastic support to us for 10 years on this project, and we're very appreciative of that. They have funded some of the other work here, which is linked in part to our work. There's a project looking at proteins, proteomics it's called, running out of Oxford, which they funded and uses our samples. There's a project, and clinical data, there's a project looking at MRI scans run out of Nottingham, which is tied into our study and involves a subset of people in our study and some other people. There's a project looking at pain specifically run out of Manchester. I could go on. We're very cross-linked to a lot of other projects, and the data in its entirety has been enormously helpful in just answering our own research questions, but also in helping other groups. You see some other countries mentioned there. The French have got a project more recently set up called Precise PD, and they've linked across with us to learn what we've been doing. And the idea is that we will share data over time. So at that, which is just about the hour, I will stop, and I'm happy to take questions. I think we're going to have a break first of all.
Yes, thank you very much, Donald. That was a really fascinating talk and covered such a wide variety of topics. I've really been looking forward to hearing from you and more from the Tracking Parkinson study. So yeah, on behalf of the Greater Glasgow Research Interest Group, thank you. Yes, and you're correct. It is time for a 10-minute comfort break. Whilst we're on break, everyone please be thinking of the questions you'd like to ask Donald. And once we get back, we'll open the Q&A session, but feel free to type your questions into the chat box, and we'll start those promptly at 11 minutes past 4. So see you in 10 minutes time.
There's actually a lot more detail to that. It depends if someone's on a drug that's longer acting, they'll have to leave it off for longer than a short acting drug. So all of this was factored in, and what they were to do the following morning was also defined to make it standardized. So yeah, that was a task that we did to make it a standardized thing rather than have the conditions being responsible for the variation.
Thank you. I have another question here from Tim King of the North of Scotland Research Interest Group. In the UK data set, has it been possible to identify regional differences in Parkinson's type that might suggest an environmental influence, for example, urban versus rural living?
No, not really, because although we actually did have an environmental risk questionnaire included in our project, the sort of design we have wasn't sufficient, or it wasn't designed to answer the environmental question, which requires really quite a different design. In fact, on a bigger scale and with more depth about environment. So we haven't really set out to do that, and haven't tested for that. That would actually be quite a different project. So although I've said that the current project can answer so many questions, actually it can't answer urban versus rural. It's just not set up to do that. It would need actually more participants, but would not need to collect serial data in the same fashion. But so these large-scale population-based studies would be the way to go about that. I'm enthusiastic about large-scale population-based studies involving Parkinson's. I think more could be done with that, and I would like, in an ideal world, to have a kind of register of everyone with Parkinson's and in the whole country. But one can always dream about such achievements.
Thank you. Now, I'm aware that you won't be able to really comment on people's individual cases or give any medical advice. So do say if these questions, if it's not possible to answer them here. But someone writes, I have a gene mutation of MYBPC3 causing hypertrophic cardiomyopathy. I was also diagnosed with Parkinson's at the same age of 52. Alpha-synuclein seems to feature significantly in the HCM gene mutation. Is it possible there may be an as yet undiscovered link? And do you think it would be beneficial to undergo further genetic testing?
Well, I think it's possible that there's a link because, as you say, Helga, there's a link there with alpha-synuclein. Hypertrophic cardiomyopathy is much rarer than Parkinson's, and yet in your case, and with the relatively young onset of Parkinson's at the age of 52, there could be a linkage here that is as yet undiscovered. As to the question of whether it would be beneficial to undergo further genetic testing, first of all, in terms of an alpha-synuclein gene being the source, genetic mutations in alpha-synuclein itself are extraordinarily rare. And we didn't, in our whole project, we didn't have anybody with an alpha-synuclein gene mutation at all. So it's the rarest, amongst the rare thing, it's really, really rare. I think there's always this question about what that means about would it be beneficial. I mean, it wouldn't turn a corner as regards choosing a treatment for the Parkinson's. The Parkinson's will be treated the same whether something is found in relation to alpha-synuclein or not at the present time. So in the sense of finding new treatment for you, it wouldn't be helpful. In terms of scientific discovery, it would be potentially interesting. But I have to say, I've not really looked at the hypertrophic cardiomyopathy element at all yet in our work. It's something that we could look at. So you flagged up something of interest, and one of the things we would like to do, we will at the moment we've tested a lot of the material for genes relevant to Parkinson's, but we haven't tested for wider genes. So we've looked at thousands of genes, but there are millions of genes in the human body. So one of the things we're wanting to do and trying to set up is spend even more money and analyze the whole gene, the whole genome. And from that, we could find these kind of things like the linkage to this gene linked to the hypertrophic cardiomyopathy by having a kind of whole gene approach to things. At the moment, we've not actually had a whole gene approach. We've had a targeted approach of things that cause neurological problems. And obviously hypertrophic cardiomyopathy is a heart problem. So we've not really studied that yet, but we could do, and we would hope that we will do if we get this extra arrangement made, which we're trying to set up, that we'll get funding in. It's going to cost about, it's in euros, but it's about 2 million euros to do this, just the laboratory work to do this and find out all these other genes.
Thanks, Donald. There's a few other questions around, is it possible for people with younger onset Parkinson's to find out if any of the genetic variations apply to them? And, if you think genetic testing is something that people should be pursuing?
Well, again, I mean, first of all, there is a facility to have this tested on the NHS if there is young onset Parkinson's. I think I think it's set at 45 years and less, but that is available on the NHS. Back to my earlier comment about is it worth doing? It depends. It depends what you mean by worth doing. I mean, as a clinical researcher, I'm always interested in this. So I think it's interesting. And as a person with Parkinson's, you might find it interesting. But the question is, at the moment, would it change the treatment approach that you get? Not yet. Might it do in future years? Yes, probably. That was one of the themes of my presentation, that knowing the genetic underlying cause could become relevant. But you could take the practical approach and say, well, until there's a treatment related to a genetic type specifically, there isn't a great advantage in knowing. It's a personal decision.
Thank you. Let's see, lots of questions coming in. There's a question here from Jamie. Does Parkinson's affect the bladder, causing it to weaken? And are there any remedies or medications that could help if this is the case?
Yes and yes, briefly. Bladder symptoms are quite common with Parkinson's, and there are bladder stabilizing treatments available. As like all medications, there's pros and cons to taking such treatment. So in a broad way, the answer is yes and yes.
Thank you. Jane asks if you could elaborate on the pain study at Manchester that you mentioned, and if the pain, is the pain related to rigidity, or does pain come from another source?
Yeah, so this was the purpose of the study done in Manchester, or coordinated from Manchester, and it involved people with Parkinson's in our study as well as the Oxford Discovery study that I mentioned. So it bridged both studies. And one of the ideas was that pain is not just one thing in people with Parkinson's and will come from different sources. So some of it is related to rigidity, and ibuprofen as an anti-inflammatory can help, and simple painkillers like paracetamol can help. It's actually, when we look at how many people with Parkinson's take fairly regular simple painkiller like paracetamol, it's quite high proportion. But what the Manchester group defined really was that there's also, the brain receives a pain signal and processes that and really recognizes it as pain, and puts a severity rating on it, if you like. And it seems that in Parkinson's, the processing of that isn't quite right. Or putting it another way, some people with Parkinson's, their brain is telling them that it is more painful than it would do if they didn't have Parkinson's because of processing. Now, you could use ordinary painkillers, certainly. Like all medications, pros and cons of it. Too much ibuprofen can irritate the stomach and cause stomach ulcers, for example. But reasonable use of it that is helpful is good. The pain study is saying, well, could there be a treatment developed that helps sort out this processing signaling thing in Parkinson's? And that's the kind of line that they're taking. So that's very early stages of research, but kind of very novel approach to say it's not actually as painful as it need to be if we can modulate the signal.
Brilliant. So Colin asks, what do you think the future looks like for people with Parkinson's, and are there any major breakthroughs on the way? And also, if you have sufficient funding for your research program, and how can the Parkinson's community support?
Well, I've mentioned the huge investment made. I mean, before the Sergey Brin contribution, the Michael J. Fox Foundation has been very important internationally in increasing investment in Parkinson's research. I mean, Parkinson's UK is a huge contributor to Parkinson's research, but I've mentioned before how so much is international these days. So it's very important that we've got different lines of research going on in different continents. So there are more research studies in Parkinson's and more money being spent in Parkinson's than ever before. So it is a positive time. And are there going to be breakthroughs? Yes. When are they going to happen? Don't know. We're at a stage of, I looked about a year ago at how many current projects that this is new studies, new treatments that potentially could come through, and the figure was well over 100 possible new treatments. Now, treatments fall at different points. They fail for various reasons, but some of these will succeed. It's inevitable. There's so much being done, and so much novel thinking, and so much careful targeting. Some of this will turn positive, I'm sure. We'll get new treatments, and they will make a difference and help people. The other part of the question was, do I have enough funding for my research? I mean, any researcher will always say, I could do more with more funding. Okay, so I'll just say that. Everyone would always say that. I mean, at the moment, my big target is to try and get this €2 million type funding for the bigger genetic analysis of the whole gene. You know, we've taken a blood sample, we've stored it in duplicate, so that if the place where our primary store goes up in flames or something, we've got a separate store, and you know, we've got one store in Wales and one in England. Deliberately, we've got a backup store in another site in England, so everything's in duplicate because it's so valuable. And one of the things we want to do is this whole genome analysis, which would cost about €2 million, but I think that would be great to get. We haven't asked Parkinson's UK for that because we think there's another way of doing that, and you know, that it will take a little while before we know whether we'll get that funding or not.
I think you've partially answered another question here, which is, what is the next area of Parkinson's research for your team, Dr. Grosset? And also, once the tracking Parkinson's study concludes, what's next for the data gathered and the results?
Well, at the end of the project, or I mean, even on an ongoing basis, we're sharing. So we get requests to share our data or a subset of our data from different places, and we give that to people. So we share our data already, and we share our samples. So I did mention briefly the Oxford study, which is looking at protein patterns. So that dips into our samples. So when we got people to give us a sample every 18 months, we did a lab processing thing and created 6 so we took a very small amount of blood, but then we split it 6 ways. So we can take out and analyze 6 different things, or we can do 6 different projects, but we can actually even do better than that, because we can do an unfreeze and a refreeze. We can thaw it and refreeze for certain projects. So we can get even more than 6 projects done. And often these projects require just a very tiny volume to do measurements. So we hope that we will be a resource for projects for many years after the main project is completed.
Thank you. Another question here asking, looking at your study data, the response figures to levodopa are presumably averages for each individual. Have you looked at the degree of variation in response within each individual, i.e., why is it that my response can be quite good one day and very poor the next? And does everyone with Parkinson's have the same range in response from day to day?
Well, we haven't really done that because we just took this this response we did as a one-off in our project, and I said we did that in about 1,000 people, and they needed to be on L-DOPA-based treatment. But actually, for some of those that hadn't done it at that first time point, which was at 2 years, later on when they were on their L-DOPA treatment, we did it. But what we've done actually is, and I've talked quite a bit about how our study is helping others, so other people come to us, but we go to other studies as well, and we say to them, we're analysing this, can we access your data or your samples and combine? And one of the other projects is a US study, which is smaller in scale, but has been running a bit longer. It's about 400 people. And we've been looking at their data because they've done more of what they've asked here by Jarlath. They've repeated the challenge test many times, up to 10 times in an individual. And we've been analysing that to try and answer the kind of question that's raised here. And that analysis work is ongoing, but it's a fantastic resource, and the American study makes that data available to us on request. So we're in the midst of analysing that. It won't get down to a day-to-day variation answer, but it will get down to a variation over time answer because they're repeating it every 6 months. So I think it is one of the mysteries as to, it's a defining feature of Parkinson's, as I said earlier, this variation one day to the next. A lot of brain phenomena are cyclical, and I think Parkinson's fits with that. As to why, I don't know. But if we consider episodic events in the brain, episodic migrainous headaches, episodic epileptic seizures, you know, these are the epilepsy, quite dramatic variation in behavior. But why does it just spark off at a certain time? You know, I think we just don't understand quite a lot of this fluctuation that goes on in the brain, and it must cross a threshold for reasons that we're not really understanding, and just causes a good or bad day in Parkinson's, or a day with many seizures or no seizures in people with epilepsy, and so on.
I think it's really positive to hear about all of the collaboration and data sharing that's happening internationally amongst researchers. I think we always like to hear that studies are working together and sharing their findings. A question from Mark Brown. Any of the research findings from your study, are they being put into practice to help people with Parkinson's now?
Well, in a sense, yes. I mean, so one of the things, and I haven't touched on it today, one of the things we were interested in was if the person with Parkinson's had problems with their circulation, by which I mean blood supply to the brain, and in particular the deep brain areas that control movement. And in its fully developed form, this is a stroke disease causing stroke events and mini strokes. But there is a sort of more slow process of sludging up of blood vessels and so on, which can cause trouble. And we did a large piece of work analysing the participants in our study and showing that this issue was relevant to consideration of treating things like cholesterol. And we passed that message out to the Parkinson's community, both the clinical and the patient groups. So what we showed was that this was being rather neglected, and that more attention ought to be paid to it because it was relevant to people's walking and to their memory. So that's not a new treatment for Parkinson's, but it's an important approach to saying don't just assume that memory problem and walking problems are down to Parkinson's. Don't neglect the detail about things like circulation, and that I think has been picked up and is a more active treatment approach that is implemented these days, rather than just accepting that everything is a Parkinson problem. So it's indirect but beneficial to people with Parkinson's today.
Thank you. And 2 questions on current events. What has been the impact of COVID-19 on your research, and I suppose Parkinson's research generally? And also, will the UK's departure from the European Union affect collaboration with other European countries and researchers?
Okay, so the effect of COVID on research has been significant. There's been a pause in some studies that were about to start up, some clinical research trials. In terms of our own studies, for example, there was a pause on visits, and then there's been a replacement of quite a lot of what happened face-to-face with telephone and video. And then, just in the interest of keeping face-to-face time down, so we do as much as possible by remote, and then we can see people very briefly for a blood sample, for example. So these are effects on the research. I mean, just sitting with somebody with Parkinson's and their family for an hour to do the research doesn't seem wise in the COVID climate. So we've modified it, and we don't use Zoom, but we use similar stuff. The UK and the European Union, we don't know yet exactly how it's going to impact. I mean, we're already collaborating at the European and US level, US, North America really, because Canada is quite strong in the Parkinson's research arena. So we're able to cross borders with our research. We're not particularly dependent on being a member of the EU to be able to do things. So I don't see it as hugely problematic to my line of research. I think there is another negative angle though, which is that at the European level, there's a lot of funding that comes that is offered to a minimum of a certain number of countries collaborating. So it might say a minimum of 3 European countries will collaborate, and they apply to EU central funding for medical and scientific research. And not being a member of the EU, we won't be able to access that funding, obviously, in the UK. Now, the UK government have said, oh, we'll match said funding. And it remains to be seen how they do that, because it's slightly difficult to conceive of joining, let's say, 4 EU countries in an application to the EU and saying the British arm of this would be funded independently by the UK government. It could happen, but there's a slight sense that that's not just so clear cut as just being a member of the club rather than being an outsider, that we might fall off the radar, or just the extra bureaucracy of it would cause trouble. Time will tell. I'm not wholly negative about it, but on the EU funding, EU projects, and there is a lot, hundreds of millions of euros go to medical and scientific research from EU budgets. That is a concern to many of my colleagues who have got such funding historically.
Interesting and challenging times we live in. A question I missed from the chat box from Mark Riley. What are current thoughts on a gene therapy delivery method?
I'm just trying to put some more light into this. Yeah. Right, gene delivery. It's a stuttered program really so far. So the concept could be that if you're running short of a chemical in the brain, that you could run in some treatment that influences a gene and ramp up the output of the chemical. That's one concept. It's not really managed to achieve positive results so far. I think it's got some potential, but I think that the other approaches targeting these proteins at the moment seem to be more likely to succeed. That would be my guess.
Okay, and two questions now from the members of the Greater Glasgow Research Interest Group. Are you aware of any new research opportunities or investment coming to the west of Scotland, or I suppose Scotland more broadly? And are there any current or sort of upcoming opportunities for people to take part in any clinical research across Scotland, particularly in the west?
Well, there's a couple of the earlier questions have been about getting a gene test done, and there is a project which is being led in Glasgow by one of my consultant colleagues, Vicky Marshall, which is a European project and takes a spot blood sample, meaning it's like a finger stab that you would do, and put that in the post, and it gets posted to Germany, and they do a genetic test. And that's a European project, but with penciled in, as I understand it, at least 3 Scottish sites, including Glasgow, and Dundee are the 2 I remember. I think Edinburgh's probably involved as well. So that's a project where there is this desire to understand even more about the prevalence of these genetic variants in the Parkinson community, and really underpinning that is the future plan to then target treatments to people with these genetic variations. In terms of new drug trials, there is a pipeline of potential new drugs. This is currently on pause for new projects really in Parkinson's because of COVID, but will restart at some point. And I think there will be West of Scotland participation in those in due course.
Excellent. I've popped a link in the chat box as well for the Parkinson's UK Take Part Hub, where people can enter their postcode and search for studies that are recruiting participants, the majority of which will be things you can take part in from home. But we'll keep that up to date. We're coming to the end of the Q&A. So one final question from the chat box. Are you and your colleagues lobbying the UK and indeed the Scottish government on funding for the post-Brexit landscape?
I don't know at the Scottish level. I've personally not been involved, but I think my research scientist colleagues have been. I know that there's been lobbying at the kind of UK national level about this. Quite a bit of this funding is actually UK funding at the moment, rather than just Scottish funding. So the 2 big funders are the Wellcome Trust and the Medical Research Council. So these are UK, and the money is UK money rather than Scottish money. So there's been a lot of representation by clinical scientists and researchers to the UK, and the response from the UK government has been, as I said, that they will match funding and that they would administer it through the likes of the Medical Research Council, MRC, which is good. So yeah, there's been lobbying on that.
That's great. And Annie, the Scotland director of Parkinson's UK, has also pointed out that Parkinson's UK are lobbying through AMRC as well, and there are Scottish case studies included.
Yeah.
I'd like to ask this event being the launch of the Greater Glasgow Research Interest Group for people affected by Parkinson's and researchers, healthcare professionals to come together and promote participation in Parkinson's research. What advice would you have for the group, or what would you like to see the group achieve in the coming years, or how could the group support you in what you're doing?
Well, I think, first of all, there are projects come up of different types, and the traditional way in which we invite participation in projects is by people who come to clinics, and that's fine. But I think that a group such as this one can be a hub for spreading information about projects, and in a way, I don't mean bypassing it, it's supplementing the personal approach in the clinic. So I think it's and the size of it is very impressive, in terms of the number of people joining today and so on. So and the website, these are the ways in which communication can happen rapidly in today's world, and can keep people up to date, and can get people actively involved. But I also think that you can be, you can watch what's happening in the research world with Parkinson's very well. For example, through the Parkinson's UK newsletters and literature, there's very good little summaries produced of projects, and I mean, either on the website or if you get posted out a newsletter, to keep up to date with what's happening. So you can be both active personal participant in projects, but also actively interested in just soaking it all in as well.
Thanks, Donald. I think that brings us to the end of the time we've allowed for the question and answer session. So thank you so much for answering all of our questions so openly. I hope we haven't missed too many questions from folks. Colin, Chair of the Greater Glasgow Research Interest Group, I wonder, before we hand over to Annie MacLeod for some closing reflections, if there's anything you'd like to say or ask.
It's been really boring, hasn't it? Can you hear me?
Yes, loud and clear.
It's like a masterclass. Donald, I was fascinated. And I need to listen to that recording again to really understand more of what you've been saying. But I thought it was a superb way of introducing us to the complexities of the work you're doing, but also the excitement that you show in your presentation. So thank you so much for sharing your time and your expertise with us. And we look forward to linking with you on other developments as time comes by. One of the things we started frustrated with in the new Greg is not knowing who's doing what, where, and when. And we assume there's a network of people like yourselves who know exactly what's going on in the west of Scotland. And if there's a way of linking us into that network, we'd be really appreciative. Because I think the people who attended today may be interested in participating in trials themselves, or certainly knowing more about what's going on locally. And I think we can develop a good partnership between us with people who live and work in this part of the world and your colleagues in the field. So if there's a way of helping us to understand how those networks operate, we'd be really grateful. I should say, Liz, that part of our development as a group has been highly supported by our friends in Dundee and Edinburgh. And through this Zoom meeting, I think we should say well done to David in Edinburgh with a colossal response to his recent conference. So well done, David. We're pleased to be part of the Scottish network. Apart from what's going to happen in Wales on Saturday, when we take on the Scots at Llanelli, there may be a bit of a variance in mood and temper, but we'll live through that. But on a serious level, it's been fascinating having you, Donald. I appreciate the time you've given. I don't know how you sleep carrying all this knowledge with you and putting all those permutations together. I know you can't see the people participating today, but I'd like to think that we can do what we did for the nurses and the doctors, is to show appreciation in the usual way. But before I ask people to do that, Eric has been very quiet today, And I'd like to combine Eric, who's been the master of our website development, and to say to people who are with us today, we'd love to get comments from you about how useful you found the website and how you'd like to see it improved. It's a brand new initiative and it's been taken on by people who've given their time so kindly on a voluntary basis. So I'm really excited about the opportunities presented as a result of today. And you know how much I appreciate the help from people in Parkinson themselves and the team in Glasgow. One last thing, if I may, we're looking for people, particularly from the local groups, to join us as research champions. It doesn't mean to say you have to come to more meetings if you don't want to, but it'd be good to know that within every group, there's someone who takes a particular interest in what's going on in the research. So because we've got someone to feed information to and from, so we know the groups are part of the wider debate. So if people are listening today, it'd be good to know that that's going to happen. So to all of you who spent time today with us, thank you so much. We should say something. Maybe I'll ask Donald to do this. Donald, do you want to officially launch the start of the Greater Glasgow Research Interest Group? So we've got a time and place when this happened for future references, and also to launch the website officially as a tribute to all those who've been involved, and particularly Eric. So there are two linked and important developments which I would like to invite you to officially launch on behalf of the team, and to see the development of a new structure in Glasgow, which we hope will be helpful to everyone. Thank you so much.
I should have sent you some big scissors for a ribbon cutting.
If Donald could officially launch, it'd be great.
Yes, well, thank you very much. I'm honoured to do this, and if I had known this in advance, I would have. I wasn't thinking of cutting a ribbon. I was thinking of opening a bottle.
That'll do.
Yes, we can do. Let's do it afterwards, because I'm not prepared. But absolutely great to have this group active and part of a network, and the website, as I say, is a way to share info at breakneck speed and I see the comment about more involvement. Absolutely, we were dependent so much on everybody who participated in that project and all the other projects that we've done, and many on this call. And I've had to scroll through again the 3 pages of people, and I see so many names and even some faces of those on video that I know and have known for many years. So wonderful group this. I wish you all the best with it, and I hereby officially at 4:52 on 27th October pronounce the group active and alive and open. Yippee!
Thank you. Before everyone rushes off to get their dinner ready, I'd like to welcome Annie McLeod, who's the Parkinson's UK Country Director for Scotland. She has some important closing reflections for us.
Well, I don't know how to follow that, Donald, but what an absolute privilege it's been this afternoon to listen and to hear and to join some new faces, some familiar faces, and actually provide everyone with hope. And I think one of the key, in closing reflections, the science aside, the biggest word that comes to my mind is collaboration. The collaboration that you've shared of how the data is so fundamental for studies right across the world and funded and taking part here in Glasgow and across the UK. And I think collaboration, because our research strategy at Parkinson's UK is designed by people affected by Parkinson's and has informed every step of the way through the research network. I can see Tim there. How we award our grants, all the rest of it is all designed as people with the Parkinson's community. So your involvement and your leadership with us, we are in it together, and we have to work together in order to make Parkinson's less significant to all of us and get these new treatments on the table. I think in the times of COVID, it's really important to point out that you know, we are all our grant programs have been awarded this year. Our funding, as we've planned, has continued to do so. And that's because of the generosity of people like yourselves, who know that it is the one thing that we can do to, well, there are many things we can do, but it's such an important use of money to keep those studies going. And I think, you know, it's interesting, I'm often asked when I'm out and about of why isn't everything that's funded in Scotland, or it's money raised in Scotland, spent in Scotland. I think Donald's explained that so well this afternoon about how it's a European, an international, a global effort to really bring those treatments to the fore for this condition. So I think, you know, that is there. What I would say, not that I'm competitive, is that there is 15 million quid's worth of money invested in Scotland at this time. We are I think, leaders in terms of how we work with the Parkinson's community and the research community and bring that collaboration together. So I think on a Dreich afternoon, it's been hugely uplifting, Donald, hugely uplifting, Colin, Eric, and all the team. Chloe, Liz, thank you so much, and thank you very much to everyone for joining. And I look forward very much to the next GRIG meeting, and we've got DRIG meetings. And I suppose the one thing about this COVID nonsense is that actually we're able to spread the word right across Scotland and beyond, and really nice to see someone joining from Ireland. So long may that continue, and thank you very much. So I wish you a good bottle opening for the launch of the group, and we hope to see you all very soon.
Thank you, Annie.
Thank you, Annie.
Over to you, Colin, for our official close.
Oh, the official close is to say thank you to you and your colleagues. You've been tremendous support. We couldn't have got there without you. I'm glad that the other Colin Williams is listening in and sending messages, and I know how much we appreciate everything they've done. And we've got a super team in Glasgow. We'll be very happy if other people would like to join us, particularly to be research champions. So the door is wide open. We may not be busy until after Christmas, but we're certainly going to use Eric's website to promote what we do, to encourage you to participate yourselves, and to form links with other people undertaking the research in Glasgow. We're really keen to be partners in the development of new ideas, new solutions. We'll share the frustrations, we'll share the hopes and the aspirations too. And finally, to say again to Donald, it really was a masterclass. Thank you for your time and your commitment. It's been an inspiration. Have a very good evening. Thanks very much indeed. Goodbye for now.